So, sometimes we do something one lab period and we follow it up the next lab period. So one of the things that we'll do today, right, is we'll look at our streak plates from last week. And the main question we're asking is, do you have isolated colonies? And as I said last week, you're not always necessarily going to get that your first attempt. If you made any notes last time, like you made a mistake, right? You know where you went wrong. Like on today's quiz, right, there was one plate that I asked you, what do you think went wrong? Okay. Any guesses as to what went wrong with that plate?
They didn't do the quadrants right.
They didn't do the quadrants right.
And they didn't take from one.
Yeah, it clearly didn't look like they took from one section to another, right? It looked like they did each one individually. And in fact, that's what they did. They took from the culture and put some in each one of those 4 squares. They didn't follow directions at all. Right? And so sometimes you can kind of figure out by the way it looks what may have gone wrong, but not always. And sometimes when you make a mistake, you know you made the mistake. And as I said, you should document what you forgot to do or what you did wrong, right? That's what a good scientist would do. So each one of those little colonies, after about 24 hours, grown at 37 degrees, one of the cultures that we grew, in a liquid culture with aeration, if we had a shaking incubator, right, if we were able to shake that up, E. coli, for instance, Escherichia coli, can double every 20 minutes under ideal conditions. Good temperature of 37, good food supply, that good aeration. Let's say on an agar plate it would grow a little bit slower, and again that's because you're on a surface and you're piling up on each other. Imagine that pile, and nutrients and is not as readily available if you're in that middle of that pile as opposed on the edge, right? So a little bit slower rate. So suppose the doubling time is about 30 minutes on our plates. After 24 hours, if you started with a single cell, you'd have 48 doublings, right? Where it's copied itself 48 times. You've gone from 1 cell to 2 cells, right? But this goes up exponentially over time because 1 cell becomes 2, 2 become 4, right? And then we have 8, 16, and on upwards. So if you do 2 to the 48th power, that's a pretty big number, right? If we keep doubling, we doubled 48 times. So it would be equivalent to, another way you could write it is, say, approximately 50, right? We could easily divide it up to 2 to the 10 and then times it by 5. So 2 to the 10 is 1,024, right? And then multiply it, so say that's around 1,000, and then multiply it by a magnitude of 5, right? So 3 times 5, because we wrote in exponents. We're talking about to the 15th power. So we're talking about 15 zeros. So that's 1,000,000 times 1,000,000,000 cells. That's a lot, right? That's a lot. And when you guys look at your colonies, there's like little tiny dots even, right? So maybe off by a factor of 3.5, but give you some idea. So this also depends on what organisms you're working with, right? We used Escherichia coli as an example here, and we will use Escherichia coli later on in the semester and actually count the number of cells in an original suspension. And Peter's trying to devise it so we can actually figure out about how many are in a colony. And so he's done some research that he's summarized here in one of our later slides. And as I said, growth is going to vary because of the environment and how they're arranged on that plate, right? What they have available to them. So this is from a paper, and so an ordinary colony of E. coli, 37 degrees after 24 hours, expect about 3.3 times 10 to the 9th growth, right? So again, about 1 billion cells in that little tiny speck that you're seeing on those plates. Suggests that the double time is probably around 45 minutes on average for that actual group of cells. So, can we experimentally determine this? Yes, and we will try and work this out, right, for you guys this semester. Something new that you guys are going to get a chance to be part of. So, and this is a link to the a video of E. coli growing and dividing. Pretty cool. So, you know, our experiments are even kind of linked in this course. And what you guys are going to be learning right now, after midterm, you're going to be doing these stains on an unknown organism, right? So it's important to learn these procedures and then be able to perform them again later to be able to determine what unknown organism you've been assigned. So one of the things that helps us in identification is the shape of the organisms and how they're arranged in three-dimensional space. So why do we actually stain? One of the reasons is to create contrast. These bacteria would blend in with their environment if we didn't stain them. So they're mostly translucent. So we use dyes so that we can actually visualize them under the microscope. There's one problem with this. Most of the dyes that we use is going to do what to the organism? It'll kill them. It will kill them. Right? So we're not looking at live organisms. But it's going to allow us to see things like morphology. Right? So what's the shape? When we look at them on the plates, right, one of the things we looked at was the shape of the colonies. Are they round? Are they ruffled? We saw that ruffled one under the dissecting scope. And also the individual cells, when we look them at under the microscope, how are they shaped? How are they arranged next to each other? Another thing that is important for cells is surface to volume ratios. So, and this is dependent too on the shape of the cell. So we're going to do a little experiment with Play-Doh I stole from my son. Right? We're going to make some bacterial shapes and make some comparisons. If you have stuff that's of equal size, right? Like if we have a ball of Play-Doh that we're going to do, and we have it equal size, but we can make different shapes. If you were to calculate the surface area to that actual volume, there's a distinctive difference between rod-shaped, what we will call bacillus, and cocci, which are the spherical shaped. But where this comes into being important for cells is what happens at the surface. Anyone know what happens at the surface of the cells? These are unicellular organisms, right? They're a single cell. But what needs to happen at that surface? Remember, a membrane is semi-permeable. So stuff is going in and stuff is coming out, right? Nutrients are coming in, wastes are coming out. This happens through the protein pores, right? And some things can diffuse right through the phospholipid membrane. But a lot of them are controlled by these protein pores, these proteins embedded in the membrane. So the greatest surface area you have, the more proteins you could have and the more exchange with your environment, right? So in this case, a greater surface area compared to your volume would be advantageous because you could do more exchange with your environment. But it depends on the type of environment, right? So we'll talk about different types of environments. So this can limit to their size, and this is part of the reason why scientists believe cocci are so tiny, because they tend to, on average, if they got really big, their surface to volume ratio doesn't increase as much as a rod-shaped bacteria would. In comparison, they have less surface area to their volume because of their shape. Okay? So because of that, they can only get so big. If they get really big, they don't have enough surface area to support all that volume on the inside. So as I said, today we're going to stain. And there's two basic chemical formulas, you could say, or types of dyes that we use. Basic stains are the most widely used. And this means they consume hydrogen ion protons. They become positively charged. And therefore the environment will tend to become negatively charged. Our most cells that you work with, including our own cells, have an overall negative charge to their membranes, and especially to the inside. Your DNA is a negatively charged molecule. So these positively charged dyes are attracted to the cells, right? And that's why they stain them. This opposite in charges, right? The basic dyes are positive, our cells are negative. That opposites attract. Heat fixing we will do for one of our staining procedures. And as the name implies, we're going to use heat to fix, to literally stick them to the slide, right? So back in the day before non-stick frying pans, if you threw an egg in a pan, what would happen to it? It would stick. And it's those proteins that coagulate, right, when you fry an egg that really become sticky. Same thing with cells. There's a lot of proteins in cells. When you heat them up, they become sticky and tacky, and they will stick to our glass slides. Now, of course, at home we have our non-stick pans, or in the old days you would put some type of grease, right, to keep it from sticking. In this case, we want it to stick because we want it to adhere to the slide. So when we're rinsing our stain off, it doesn't wash off the bacteria. They stay stuck to the slide. Now, of course, some of them would stay stuck even if you didn't heat fix, if you let them dry, but not as many as if you heat fix. The other important thing is, what does heating typically do to bacteria? It'll kill them. So it's a twofold thing. We will kill them, and we will stick them to our slides, right? So that they will come out good. And so I'll show you guys this procedure today, and you will do that as well. When it comes to the individual dyes themselves, there are different components of the dyes. So the chromogen is the actual color component of the dye. And they're usually these cyclic molecules, right, carbon in a circle. Planar, meaning that they're, within a plane. Aromatic, right, they tend to be volatile, smelly. And light is going to affect them and produce a particular color, right? So when the light hits this molecule, it refracts and shows us a particular color. The chromophore is the region of the dye that gives it its particular color. So again, it's related to the chromogen, hence the chromo part of it. The oxychrome, according to your lab manual, refers to the charge region of the dye molecule that is binding ionically to our cells, right? So that's the part that becomes positively charged when we're talking about basic dyes. According to Wikipedia, which isn't always necessarily a good resource, right? But believe it or not, they do get some things right. And you should always, if you're using Wikipedia, go to their original resources and double check. It's almost like reading someone else's term paper when you go to Wikipedia. The chromophore alters both the wavelength and intensity of the absorption. The word chromophore comes from the Greek root to increase, right? So in this case, it's helping us to increase contrast, allow us to see. Chrome meaning color. So this is Peter's note for us. He feels that it is unnecessary to determine the color producing and binding regions of a dye, and I feel the same way, right? So this is pretty much all we're going to talk about this. Just know that that's what they're talking about, right? Is how the different components of the dyes are associated. So give you an example of some of the dyes we use. And for some people, having a picture to represent things. So remember I said ring-like structures and become positively charged. So this is a very commonly used dye that you guys are going to use today. It's crystal violet. It's a basic dye, right? It becomes positively charged in solution. And we use it, we're going to use it in our simple staining procedure today. It's also the primary stain when you use the gram stain, right? It gives the overall purple color. So malachite green is something we're going to use later, right? And as you can see, it's a greenish dye. And look, it's structurally very similar, right, to crystal violet. Again, positively charged dye. And the differences here, right, What is the only difference between these molecules? What alters the light absorption? In the case, it's this portion of the molecule. And resonance is a chemical interaction, and it has to do with the conductivity of light. And you use this a lot in chemistry class, right, when you're looking at molecular configurations. So for this guy, he goes through a change, right, and this is the interactions, the resonance that happens, almost like a vibration you can think of, that generates the color. So the frequency of the electron movement is what absorbs and then emits the color that we see. Kind of cool stuff. So it absorbs the light here. It actually absorbs red light, but it's going to reflect back green light to us. So one of the things like chloroplasts, when you look at green plants, they're actually not absorbing the green light. They're absorbing the other light spectrums, and they're bouncing the green back to you, and that's why they appear green. So for crystal violet, it absorbs in the yellow spectrum, and so therefore we see it on the opposite side. We see the purple, that it's not absorbing. It's getting bounced back to us. So why do we use microscopy? One of the important things is it improves resolution, right? It helps us clearly see things. And you guys calculated this before, right? We have set lenses on our microscopes that have set numerical apertures. We have filters on our microscopes that allow us to use certain wavelengths of light. So we generate resolution within a limit, right? So again, we're within limits with our bacteria. They're going to look tiny, right? But we're still going to be able to see individual cells in arrangement. But really not much more detailed than that. Unlike our eukaryotes, which were much larger, right? We can see things like the nucleus and the chloroplasts and things like that. It also provides us magnification. One of the main reasons, too, for using the microscope is be able to magnify it to a level that we can see with our eyes. And we know how to calculate this as well. We use compound microscopes, so remember which two lenses do we multiply to get total magnification? Yeah, ocular, which is the ones you look through in your objectives, the ones on the resolving nose piece that we can choose between. Remember, even though you're looking to two oculars, it's only one magnification, right? That happens. The other is it improves contrast in certain situations. So there's different types of microscopes out there that can help with this other level of being able to see, which is our contrast. So we're using which type of microscopy in this lab? Bright field, dark field, phase, or fluorescence? Bright field, right? We're using bright field microscopy. So because of that, we have limitations, right? This is one of the main reasons why we stain our organisms to create contrast. Dark field microscopy, again, you're not going to usually get much contrast. That's where the background is dark and the organism is illuminated. Phase contrast, on the other hand, uses special filters to generate contrast, as the name implies. So with those types of microscopes, sometimes you don't need to stain the organisms. Or if you do, you'd be able to see greater detail, as we did in those pictures of the euglena in your lab manual. And then fluorescence, of course, as we said, employs the use of either fluorescent dyes or organisms that have the ability to fluoresce. That's usually with UV light as the energy source for that fluorescence. So again, those microscopes have to have special filters on them because UV light is damaging to our eyes. So stains can be applied broadly to determine, right, shape and morphology, right, how they look under the microscope, how they are arranged in three-dimensional space. And we can use this to learn different things about the organisms. So today, very simply, we're just going to use one dye, right? We're going to use one dye, one basic dye, and we're actually going to use two different ones. They're going to stain the cells themselves. We're also going to do another procedure, which is called negative staining, where we use an acidic dye. These guys have a negative charge, and therefore are repelled by the cells. So instead of staining the cell, we're going to stain the background, right? And that will leave the cells unstained, and we'll be able to see them as clear objects. right, to give you an idea of what they look like when you don't stain them. They're clear, right, under the microscope. But with just using one stain, that's really all you're going to see is their shape and maybe how they're arranged. Other stains that we'll do later, like the gram stain and the acid-fast stain, are ones that we call differential. And they're going to help us differentiate organisms from one another based on their properties and how they interact with these dyes. So we actually use two dyes, so we end up with two different colors, two different results when we do these stains. Endospore staining, and so these are general or specific compositions of the cell wall for gram stain and acid-fast. Acid-fast is the specific one. Endospore and capsule stains are typically what are called structural stains. They'll help us see structures if they're present, right? And so we're going to do these next couple of stains over the next couple of labs. And these are where you can see stuff either outside or inside of the cells. That stain is specifically designed to see those structures. So as I said, differential stains, right, help us differentiate characteristics about the organisms. So all of these are sometimes referred to as differential stains, although gram stain and acid-fast are the most commonly used differential, gram stain being the most popular. So this picture has a bunch of different shapes, morphologies on here, and we're going to go over the terminology that's associated with each one. As you guys wrote out in words what you're seeing, you should have also done drawings, right, of these shapes. And vice versa. You should be able to describe it in words as well as drawings. So this first one, anyone know what this one is? Cock's eye, if you're talking about all of them. If you're talking about a single one, cox us, right? So it's Latin. So us is actually singular. I is plural. Kind of the opposite of what we're used to, right? When you say I, you're talking about just yourself, right? In this case, it's a plural term. When we say us, we're usually talking about a group, right? And in this case, that's singular. So the next one, they have divided and they've stayed next to each other in pairs. We refer to this as Diplococcus. And if you're talking about just one pair, it's Diplococcus. If we're talking about all those pairs in this picture, Diplococci. You switch out the US for an I. Okay, this next one, we actually have an organism you guys work with today, and hopefully we'll see these, form these characteristic of four, right, in a square shape like this. And this is called tetrad or tetrads, if you're talking about all of them. That's a specific term to describe the individual shape and arrangement. Cocci are the only ones that can form this characteristic arrangement. The next one, you'll see they're staying together in a chain. And in this case, it's just one chain. And the prefix that means chain is strepto. And so they're cocci, so in this case, streptococcus. Right? If we had multiple chains of streptococci, right? So this one is a causing agent for an infection that most of us are aware of. Good old strep throat. Because guess what? It's caused by Streptococcus prurigens. That's the name of the organism that causes that infection. So we basically just shorten the name of the organism when we're referring to that infection when we say strep throat. How I remember, because strep is a prefix that people often confuse with the next prefix that we're going to go over, which is staphylo. How I remember it is it's a chain, and think about when you have strep throat, if someone told you to swallow a chain, you'd run away. You don't want to swallow anything, right? So think about that chain, think about your throat, right? And remember, strepto is a chain. Right? You have to tell yourself little stories and little things sometimes to remember, right? Help you remember things. That's the one that's always worked for me. The next one we have is a cubed packet of 8 cells that are cocci. And this is Sarcinia. Now you guys may have noticed some of these words you've probably heard before, just like how I said Streptococcus prurigens. Some of them are actually genus names. But we denote this differently when we're talking about a genus. We italicize them, or we underline them if we're writing them. So notice most of the time we're pretty good about the handouts and the things that we give you guys, right? That names of organisms are italicized. But as you can guess, most of the organisms in the Streptococcus genus have this characteristic morphology under the microscope before they get their name. This next one is another one that's also a genus, a very popular one that we talk about, especially in medical terms, and that's Staphylococcus. So again, in this case, staphylo referring to the clustering, coccus, those individual spheres that you see. So staph infections are typically caused by Staphylococcus aureus. Okay. So again, we're just shortening the name of the organism that's causing the particular infection. And these can be quite serious because there are strains out there that are resistant to antibiotics now. So for instance, MRSA, which is actually MRSA, which stands for Myctoline-resistant Staphylococcus aureus. Okay? So that particular strain is resistant to penicillin-like drugs that we commonly would use for staph infections. So now they switched to another drug that they commonly use, vancomycin. So guess what we have now? Vancomycin-resistant Staphylococcus aureus out there. So this battle will always continue. The bad news is we may possibly run out of antibiotics before they run out of resistance. That's the scary part, right? So the next one is what we typically refer to as a rod shape, but you can almost imagine it as a sausage or a hot dog shape, right? Three-dimensionally, right? These are three-dimensional organisms. So this one, the proper term we use is bacillus. We're talking about more than one bacilli. These guys can form pairs. So what's the prefix for pairs? Diplo. So this would be diplobacillus, or diplobacilli, referring to all of them in this picture. The next one for rods is a chain, so it would be streptobacillus. The next one are rod-shaped, but they tend to have a little bit of a curvature to them. These guys are referred to as vibrio. And in this picture, and a lot of the organisms that are in this genus also tend to have flagella. So that's what this little line is representing. This next one is a long rod that has multiple curves to it, not just one. This would be Spirilium, or Spirilia. Instead of the UM, it would be an A for plural. This picture also is showing one that has multiple flagella at both ends. This last picture is all the same organism, but they're not all the same shape, are they? No. This is what we refer to as being pleomorphic or pleomorphism. And the cell wall is usually what helps these cells hold these characteristic shapes. So there's a group of bacteria known as mycoplasma that don't have a cell wall. And so they have this pleomorphic appearance to them where they don't hold the characteristic shape. Causes an infection called walking pneumonia. Walking pneumonia is actually caused by mycoplasma pneumoniae. It's called walking pneumonia because it's a milder form of pneumonia. People usually up walking around and have no idea how sick they are, which isn't good. The last one are spirochetes, and under the microscope they look like these little ziggy lines, but three-dimensionally they're helical, right? So almost like a slinky or a spiral staircase. We unfortunately don't work with any of these spiral ones, or they're hard to grow, and a lot of them are highly pathogenic. So the bad news is, guess what? All we have is rods and cocci to look at in microbiology lab. But you guys will get excited over, successfully staining and making pretty slides. So to give you an idea, some organisms that would have this characteristic shape, so things like Lyme disease and syphilis, again, another reason why we're not working with these, right? Why we're not going to stain and look at them under the microscope. These are kind of cool. These organisms are really cool too, in that they're motile by these axial filaments, so almost like flagella, but run along the outside of the cell within an outer sheath. And so this is them in cross-section. And this allows them to corkscrew through their environment. So they literally twirl through. So we always talk about bacteria being bad, right? So this is a great aside of some research they're doing on tribal people in South America. And they're found that, and we know this nowadays, like the whole big thing about probiotics, right? And the microorganisms you have growing on your gut and in your skin can help even protect you from disease. So I've looked into the particular organisms that these individuals have and how beneficial they are for them. So, for instance, in helping preventing kidney stones, right, and some of the other problems that we experience. So this is an interesting article that he shared a summary for us. So Staphylococci I already mentioned, Staphylococci epidermis, Staphylococcus epidermidis, is commonly found on our skin, right? It's a non-pathogenic strain, right, and one of the organisms that we'll work with in lab. And so again, they tend to cluster, and these guys actually tend to produce a matrix outside of their cells that even sticks them together, even though they're unicellular organisms. So, as I talked about, good old Streptococcus priogens, and again, we're not going to work with any Streptococci. They're pathogenic, hard to grow. So you won't see any chains like this. Streptococcus pneumoniae too, right? Another one that can cause a pretty serious infections. Vibrios, as I said, is one of the ones where it's also a genus name. Vibrio cholera is probably one that you guys are familiar with. So you can see here, right, have that little bit of a curvature to them. More of a problem in other parts of the United States, but we also have the warning in all restaurants here, because if you eat shellfish, right, that may have come from contaminated waters, and you don't cook them, if you eat oysters, for instance, raw oysters, there's a potential of getting an infection like this. So, and then a couple other diseases of interest, right, caused by bacteria, right, and there's the good old, warning about the one found, the Vibrio, that's typically found in oysters. So these are hopefully similar to what we'll see under the microscope today. So in this picture, notice that the cell itself is stained. In what shape would we say this is? This is bacillus, and notice even here it's forming a chain, so that would be Streptobacillus. This dye in this picture was methylene blue because it has a bluish color. Remember, the cells themselves have a negative charge. Basic dyes have what charge? Positive charge, right? And so most of the dyes we use in lab are basic dyes. Some of the ones that we'll use today in the next couple of weeks are crystal violet, methylene blue, and safranin, which is a red dye. So we said they're positively charged, right? And so they become positively charged in solution. And so why do they stain the cell instead of the background? Yeah, cell is negative charge, dye is positively charged, the opposites attract, right? Where here in this picture, and again, notice that the cells are cocci, but they're arranged in that group before. We call that tetrads. And we do have one of those today. So acidic dyes have a negative charge. So the two acidic dyes we're going to use are nigrazine, which is this grayish black dye, and we're going to use Congo red, which is, of course, a red dye. But these guys have that negative charge, so they actually release hydrogen ions, positively charged molecules, and become negatively charged, and so they will stain the background. And this is because like charges repel. So the negative cell, the negative dye, they're going to repel each other. So what would happen if we leave our stain on too long? And the standard rule in microbiology is 1 minute. Typically 1 minute is long enough. It's not too short, it's not too long. What happens if we were to leave that on and walk away like 5 minutes later? They get too big, or they could even burst, right? So it's almost like filling up a water balloon, right? Because remember, it's going into the cell. So this would make us really hard to be able to do some of the things, the reason why we're staining it, to see those shapes and those arrangements. Especially when you're trying to determine, is it diplococci or staphylococci or tetrads. If it's just these blurry blobs, you're not going to know. Because the dye will, fill those spaces in between the cells as well. What happens if we understain? Say you only do it for like 10 seconds. You might not be able to what? You might not see them. You might not absorb enough dye that you're able to visualize them under the microscope, right? So it'd be difficulty in being able to view them. One of the main reasons why we're staining them, right? To create that contrast. So when we talk about those surface to volume ratios, I'm going to go ahead and pause mine. If we took identical volumes, right? So I'm going to have you guys take two pieces of Play-Doh around the same size, and we're going to divide them up into the two different main shapes, the cocci and the bacillus. So Peter loves the math involved, right? So figuring out the surface area to the volume and then that ratio between the two. And just taking into account a cylinder is the closest we get to a rod, right? So there are mathematical formulas that, thank goodness, you don't need to know for this class, right? It's not a geometry class. But if we were to look at, right, and so he does a bunch of different sizes, but I find our Play-Doh activity works best in showing. So how does that come into play, right? For the most part, it depends on how long that rod is, right? How much surface to volume ratio you'll have, right? So for really long rods, right, or really wide ones, like you see in this previous diagram, really wide, these are the really long guys. You're going to have different surface to volume ratios. So he's calculated a whole bunch of different examples here. In average, though, a cocci, right, a sphere, is only going to be about 3, right, surface to volume ratio. Look at these guys, 12, 10, 8. This small guy is around a sphere. But much greater surface area, right? And you guys can really see that with the Play-Doh, right? Much more surface area when you spread it out as a rod as opposed to keeping it in that ball like the cocci. And I'm skipping over that. That's Peter's math. He can explain it to you guys, to his class. So how does this affect organisms, right? Well, environment affects them as well, right? So we have this problem of surface to volume ratio. So given a particular environment, say a dry environment, who is actually going to be better suited? Well, the question is, in a dry environment, what are you worried about losing? Water, right? In a dry environment, you have less water. So, do you want a greater surface area or smaller surface area? Smaller, because you don't want to lose it. So it actually is to their benefit in a dry environment to be a cocci, right? Because they are the ones that have the smaller surface to volume ratio. Make sense? And that prevents water loss in a dry environment. They would dehydrate much slowly. What if we were in a moist environment? Who would better be adapted to that? The rod, because he doesn't have to worry about losing water, right? And so having that greater surface to volume ratio would allow it to have better interaction with its environment. Now, when we say rod too, don't forget about the Vibrio, right? And the Spirilium and the Spirochetes. They're just basically, twisted rods, right? But they're still a rod. And hence why it's a very popular shape. So negative stains, we already answered this, right? Why don't we colorize the cells? When we do negative staining, what type of dye are we using? Acidic dye, which has what charge? negative, and the cells are negative, so they repel each other. So, they give you an example of if you use eosin, which is an acidic dye, so what charge? Negative. What's it going to stain? Background, right? And if you use methylene blue, which is a basic dye, so what charge? Positive. So what's it going to stain? The cell. So what are we going to end up with? A blue cell with a red background, potentially. Remember, red and blue make purple, but you wouldn't necessarily end up with purple because of the different charges. They would stain different areas on that slide. I have to say I've never experimented with this, so I'm surprised I haven't yet. This is kind of a lead into the other staining procedure we're going to do next week, next lab period, excuse me, where we're going to combine acidic and basic dyes so that we will do just that. We will stain the cell and the background, okay, for that particular dye procedure. So we're not going to measure, but there are differences in the different procedures that we do, right? So if you were to take a Cox eye and you were to stain it doing the negative procedure and the basic staining procedure, the simple stain, you'd end up where they would appear to look different sizes, the exact same organism. And that has to do with how the procedures are. So there's a distinctive difference in these two procedures. So for simple staining with basic dyes, you're going to make a bacterial smear. So if it's not a liquid culture, you're going to put a little bit of water on your slide, and you're going to use that to mix the organisms onto the slide to create a smear. If it's a broth culture, you already have some liquid that would help you move them around. We're going to put those on our light boxes and let them air dry, and then you're going to heat fix them, right? Heating is going to cause them to what? Stick to the slide, but it's also going to cause them to shrink, right? The cells tend to shrink. So they would appear smaller when you're doing a simple stain with a basic dye. Then our negative staining, we're just going to let the dye dry. We're not going to heat fix those slides. So because we don't have that step of heat fixing, we don't distort them by the heating, and they will appear a little bit larger in contrast to basic staining or exact same cells. So the important take home there is heat will affect and essentially shrink, decrease the size of your cells. Okay. Did I hit done? I guess it takes a second.